Very good morning to all of you. At the outset, let me thank the organizers of this training program. That includes myself also, to deliver this an important, I would say, class. It's not a lecture, actually, it's a class on PCR and qPCR or real-time PCR techniques. Before I get into my slides, I just would like to know how many of you had experience with any one of these techniques in your research career. Please raise your hands. Okay, I would count around 40% students are having some experience. I am sure all of you must be having the theoretical knowledge on PCR and qPCR. Are you sure about the PCR and qPCR techniques? You are thorough with that? The basics, the principles, in particular qPCR? Raise your hands, all of you. It's only one hand is raising. So that means this lecture is thoroughly justified for this audience. Students, I would like to really tell you about the fundamental differences between PCR and qPCR. Right in qPCR, we have quite a lot of variants of qPCRs, and would like to tell you how exactly they are differing from each other. The highlight of this particular lecture would be on how to visualize the results of qPCR data. That's very, very important. I would like to just move on to the slides. I already told this morning, you keep your questions written in your piece of paper, and let us have discussion at the end of this class. And if you have more questions than the time accommodates, you can send these questions by email to me. I'll try to answer with the best of my ability, right? So that is some standing instructions. This morning I had an opportunity to update my slides. This is surprising hits I got, around 29 followed by 50,000 hits. You know what is this? If you just click the 3 letters PCR in Google and select videos, I'm not talking about all or images. This is just the videos. This is a mind-boggling number. That means I'm sure substantial number of these hits are directly connected with a technique what is called as PCR, that is polymerase chain reaction. And I just extended it to another page. I got around 185,000 hits, again videos, that is on RT-PCR. So I was just wondering, I need to have this lecture at all. You have around 185,000 videos already uploaded in Google. So then I was thinking what to do. Then I just, I didn't convince myself. I just went ahead. I put another acronym, qPCR. Then I got around 100,000 hits. That means you have plenty of online contents. Any student, anytime, with the current technologies what you have right in your hand, you can learn all the time, right? So the technologies are making the lectures and classrooms obsolete, right? So very soon we will stop doing this lecturing, making you boring, right? And in the end, you also compulsion, you have to sit here to listen to my lectures, so all those things. But nevertheless, there are more things can be explained than what you can see through videos. The purpose of this lecture is on that one. I'll try to explain what is really underneath in qPCR, right? Since PCR has become a kind of routine tools and protocol in biology research, and there is a great deal of things need to be spoken about qPCR, especially to this young audience. So the substantial part of my lecture is on qPCR, but nevertheless, the PCR will come time and again in the discussion, right? These are all the acronyms, right? I was just comparing the word biology, right? So to my surprise, none of these acronyms, any letter of this acronym is matching with biology, right? Biology has got biology, right? Isn't it? Correct? So now the point is, now PCR and qPCR has become so integrated in biology. You cannot think of a biological research bypassing this PCR and qPCR if you want to become frontline leaders in this science. So they are so intimately associated with biology since 1985. Let's put the word time, 1985. So since 1985, then the year the PCR came to limelight. In the last 30, 40 years, this technology has completely taken over the science biology. And we started learning, understanding biology much better than before. And your subject, entomology, is no exception, right? So you will very frequently find the acronyms like PCR, qPCR, RT-PCR, QRT-PCR. Right? It's all kind of, combination of letters and acronyms are created, and the literatures are loaded with this kind of information. So students, I would like to make a clear-cut distinction here. Let's see what they really mean to us. PCR, all of you must be knowing it is a polymerase chain reaction, right? And what is qPCR? The Q stands for quantitative. So quantitative PCR, right? And we have another terminology, another acronym, RT-PCR. What is RT-PCR? It's really confusing. You really don't know what the literature is going to speak about. Either it may be real-time PCR, or it may be sometimes reverse transcriptase PCR, right? So let's take RT-PCR is a common acronym for two interrelated but independent technologies, that is real-time as well as reverse transcriptase PCR. And you will also find another acronym, what is called as a QRT-PCR. This is nothing but quantitative real-time PCR. So this is something unique that we'll talk in the slides, what it really means, how it is different from other kinds of real-time PCR that will have a great deal of discussion, right? So the acronyms are well integrated in biology today, right? If you want to make a career in biology or any science in agriculture, medicine, environmental science, whatever, which deals with biological stuff, PCR has become so integrated, right? And entomology is no exceptions, right? So those who practice this kind of tools, they are standing alone in the crowd, right? And you will be rated as a top-rated scientist if you do all these things, right? So the training is all about that only, to make you one of the top-rated scientists in future, right? Let's, this lecture has got certain elements I would like to tell you. This lecture, I would like to give a brief introduction about PCR and qPCR and the history associated with that. And we have the terminologies and acronyms used, which I already told in the previous slide. And we have a history of PCR and qPCR. It's very, very important for us to know. And the major differences between PCR and qPCR, and of course, different variants of qPCR nowadays it is there. And the two of them I would like to highlight here, because those two are really dominating in qPCR research. One is the dye-based qPCR, and another one is a probe-based qPCR. So students, I must make a mention here. When I say qPCR, when I say RT-PCR, I only mean real-time PCR. Okay, you take it in that sense. I'll try to control myself in telling only qPCR, because it's the most preferred choice now. And sometimes you also forced to tell RT-PCR, the real-time PCR, in your discussion, in your presentations, right? In probe-based, you need to know a little bit about some chemistry, that is the FRET quenching and then the non-FRET quenching. That also will be on a couple of slides. I will be telling what exactly is the difference between these two. And of course, the utility of qPCR. Utility of qPCR, the acronym itself is telling you that it is a quantitative PCR. That means you can quantitate the target here. So we'll be talking about absolute quantitation. And the biggest application of real-time PCR or qPCR in biology is what is called as relative quantitation. People use this technology for gene expression studies, right? What do you mean by gene expression studies? When the plant is affected by a pest, and what and all changes happening in the plant. So if you want to study the changes at a transcriptional level, what I mean, how the plant the transcription and the protein formation is affected in the presence of the so-called insect pest. You can use this technology to know how many genes, the plant genes, are affected. Affected means its normal expression is altered. It may be either way. Sometimes the genes are overexpressed, maybe to defend the insects. Sometimes the genes are underexpressed, again to defend the insects, maybe just by what is called as down-regulating some of the genes, which the end of the day, the plant become non-palatable for the insects, primarily from the point of view of defense. So you can use this technology very elegantly in your research, what is called as relative quantitation. We'll talk much about this particular tool. And of course, a few slides, maybe a couple of slides on applications of this real-time PCR in entomology research. That also you'll see in these slides. Yeah, we'll take one by one. The quantitative polymerase chain reaction, popularly called as qPCR or RT-PCR, is a method by which the amount of the PCR product can be estimated or determined in real-time basis, right? So I was thinking the human brain, how it really works. When the PCR was invented sometime in the early 80s, and even today, if you do PCR, you really do not know something is really happening or not, right? So what you do, you mix around 6 to 7 chemicals in a tube and put the tube in different temperature regimes, and you wait for maybe a couple of hours. Then after that, again, you have to wait for another couple of hours, because you need to put them in a kind of matrix to see whether something is appearing or not, what is called as amplicons. So that means you need to spend at least 5 to 6 hours to know whether the PCR really worked or not, right? So that is the story, how the PCR, we have to wait for at least 4 to 6 hours to know the PCR setup has really worked. Now, in qPCR, those botherations are completely remote. So you can monitor what's really going on in the tube right from the clicking of your on button in the machine. What I mean to say is, you can monitor the PCR reaction right from the first cycle of your polymerase chain reaction, right? This is something called real-time monitoring of polymerase chain reaction, what is going on in the tube. So that is the fundamental difference between a conventional PCR and real-time PCR, right? So how it was made possible, let's see in the coming slides. This technique is often abbreviated as a qPCR. The Q stands for quantitative. That means the conventional PCR is not so quantitative. That's the meaning of it. And it is also referred to as real-time PCR or RT-PCR, right? qPCR does not rely on any downstream analysis. For example, electrophoresis, which I told. There is no need to perform electrophoresis to visualize the result, because from the first cycle itself, you started seeing the reaction, whether it is happening or not, right? Or the densitometry tools, just to confirm that the reactions have took place or not. That is one. And it is very versatile and enabling multiple PCR targets to be assessed simultaneously, in particular what is called the TACMAN-based qPCR that we'll see when the slide comes. It is very, very useful, which I already told you, for expression analysis, gene expression analysis, right? Whatever may be the situation. Pest plus plant, one option. You have a beneficial insect plus plant interaction. You can still study that. And of course, in other subjects like microbiology, plant pathology, we have a very big deal of applications of this qPCR. The key feature in RT-PCR is that the amplification of DNA is detected in real-time basis as the PCR is in progress, right? So you should understand how the PCR is really performed. Anybody having any doubt on that? If you don't understand how the PCR is really working, you will find it difficult, the qPCR, right? How does the PCR work? Anybody? So So there is a template DNA, that is always a double stranded DNA, right? You have a primus that is going to prime the reaction, and you have the substrate in the form of DNTPs. There are 4 species are there, right? ATCG, correct? And you have some kind of cofactors. By and large, it is a magnesium chloride, correct? And of course, a buffer will be there. Some additives may or may not be there. And you put everything together in a tube, and what you expect? And subject them to a kind of different temperatures. There is a denaturation temperature. You have an annealing temperature. There's an extension temperature. The template, which is double stranded, will become 2 single stranded. So that would attract the primus to come closer to it. Depends upon the complementarity. They come and attach somewhere. The complements are there, right? Then you have a very important enzyme, that is a polymerase enzyme. So what it does, it modulates the addition of the bases, what is called as annealing, and also the extension, right? And the single stranded will become true double stranded. This is in a nutshell. Any doubts on that? You should know this. If you don't understand this, the rest of the slides you may find it very, very difficult, right? So this is all about PCR, correct? So what I mean to say is there is a progressive multiplication. 1 become 2, 2 become 4, 4 become 8, goes and goes, right? So it is popularly what do you call, estimated as 2 power n, end of the day. The n is equal to number of cycles, correct? Isn't it? So most of the time you have used around 30 to 35 cycles, right? So that means the copies, whatever you put in the beginning of your reaction, after 35 cycles, you expect 2 power 35 number of the target, right? So just somebody can calculate what do you mean by 2 power 35? It's really an astronomical number, correct? Now, another question is, why it is only 35 to 40 times people do PCR? Why not it is 100 times? Or why not it is 10 times? Any answers for that? The answer is, whatever you are adding in PCR mix, they have a self-limiting, after some time they get depleted, correct? There would be a shortage of DNTPs because you put some fixed quantity of DNTPs, right? So no longer somebody is going to supply it permanently. So after some time, there is a shortage of DNTPs, right? Number 1. There is a shortage of the so-called all ingredients in PCR because they are all used up, right? It is spent, right? Most important point, the enzyme what you used in this PCR, it loses its power after around 30 to 35 cycles of additions of those DNTPs. So what is the meaning of it? The meaning is, after 35 to 40 cycles, right, there is a resource crunch in PCR cocktail. So it no longer can perform the polymerase chain reaction forever, right? So that's the reason it is widely accepted that, okay, around 30 to 35 times of what do you call cycling, we reached a stage where no longer the reactions can be sustained. So this is in a nutshell, correct? Whatever I told here is exactly applicable for qPCR also. There is no big difference between these two, okay? So let's move ahead. History of PCR, right? This technology came to biology, to be precise, in 1983, right? But before 1983, right in the early 1970s, the idea of in vitro multiplication of DNA has come to science. For example, 1970s, the first report of replication of single-stranded DNA is, with the help of DNA polymerase, is the proof of concept has already come to science, right? So again, 1976, an enzyme what is called as Taq DNA polymerase was discovered, right? That was again a landmark finding. And 1983, people started using this, the information which is already there in the literature, right? There is a possibility for in vitro replication. And with the discovery of this TAC enzyme, and sometime in 1983, the experimental proof for progressive accumulation or amplification of DNA, the proof of concept has come to science already, right? In 1985, what is called as the automation has come to the PCR era. That means the machines have been invented to moderate this particular reaction, right? That means before that there was no machines. So people used to shuttle between one temperature to another temperature to do PCR, right? Now those things integrated in a single machine, something like a smartphones, you have everything in a small gadget, something like that, right? In that machine, you can have any temperature of your choice, right? So that has come. And so this is exploited in this particular qPCR, right? Excitation wavelength and emission wavelength. The emission wavelength is the one which is monitored by spectrophotometer. Is it clear to all of you? I'll just pause it here. Is it clear to all of you or not? It's clear? Let's move ahead. Emission specific wavelength and excitation specific wavelength, these two things are there, right? So the question is, who is generating light in spectrophotometer? I think different options are there. You have a lamp, you have LEDs, right? Isn't it? So nowadays, LED has become very popular choice now, right? So that is depends upon the machine, right? Somebody is having lamps, somebody is having LEDs, right? Yeah, pictorially it is something like this. You have a conventional PCR, you have DNTPs, parent DNA, that's a template DNA, you have primers, you have DNA polymerase and the mixture, buffer, and all other ingredients what you put. This is a typical conventional PCR. And this is what is called as the real-time PCR mixture, right? So what is what I put in round here is the dye what is added with a normal conventional PCR. In other terms, PCR and conventional qPCR, there is no big difference, right? Only one difference, the dye is added in the PCR mixture. Is it clear to all of you? Good. So now, what is this dye? That is another question. So everything you know now, you have a buffer, you have a magnesium, you have DNTP, right? Primers, enzyme, your template DNA, plus one extra chemical. And there is a name for that. The name is detector. Why it is detector? That is going to report to you whether the amplification is happening or not. So this is called detector. In other terms, the detector is nothing but the dyes, correct? So here comes a small twist. This detector can be two different kind of setup. One is what is called as fluorescent dye. We call it as non-specific detection. Another one is a specific detection. So that means a detector is there. One is a non-specific detector. Another one is specific detector. So the rest of the slides will discuss something about this non-specific detector and specific detector, right? So the non-specific detector is nothing but a fluorescent dye, nothing more than that. You have a PCR mixture, you added the fluorescent dye. What is the most frequently used fluorescent dye? No, let's not go there. Conventional PCR, ethylene bromide, right? It's a fluorescent dye, correct? Isn't it? So you can do ethylene bromide also here. Nothing wrong in that, correct? Now, these two independent method of PCR monitoring is aptly named as dye-based quantitative PCR if you use dye directly in the PCR mixture, okay? And you have another parallel technique what is called as probe-based quantitative real-time PCR. It's slightly different from the dye-based one. In either case, you are going to add the detector in the PCR mixture. The detector can be either a fluorescent dye, right, or it can be either a detective probe. So, I introduce another word here, detective probe. It can be either a dye or it can be a detective probe. When you use a dye, this technique is called fluorescent dye-based quantitative PCR. This dye-based quantitative PCR has got some limitations that we will discuss in later. I already highlighted here. This is a little bit non-specific, right? And if you talk about a probe-based qPCR, that is by and large very specific, right? So, maybe at the end of the presentation you will appreciate the difference between these two. Let us take the first one, the dye-based qPCR. Right? You already answered ethylium bromide can be used as one of the dyes in what I call in PCR amplification, just to intercalate the dye so that you can visualize by subjecting the tube into a kind of a specific wavelength. Right? Here, the dye in question in qPCR, the most popular throughout the world, is a dye called cyber green. Right? Remember, cyber green is a generic name. There is a long chemical name is there. Please Google it out, you will understand. The dye such as cyber green 1 that intercalates in double-stranded DNA, right, are more convenient to have on hand and less expensive than probes. So, I make it another point here. Two detection methods are there. One is the dye-based one, another one is a probe-based one. When I talk about dye-based one, it is less expensive and quite easy to perform in any laboratory, right? So, of course, the disadvantage of this dye-based one is, I already highlighted, it is non-specific. Why it is non-specific? Let's take some example. Remember, cyber green is not all alone. We have number of other dyes which is there in the market. You can buy any one of them. For example, you have cyber green 2, EVA green, LC green, PIBO, YoPro, Cyto family, and the list goes, right? And of course, cyber green is the most popular one. So, what's happening here is when the cyber green is added in the PCR mixture, right? It becomes one among the ingredients in qPCR, right? And of course, the choice which I already told you, the plenty of choice, and you have some criteria to make a choice here. The dye must be thermostable, because the temperature of PCR mixture goes up to around 95 to 96 degrees Celsius during denaturation step. So, it should be thermostable. And second thing is, it should be resistant to inhibitors. So, why? Because you might handle some sample which is coming from a toxic environment. So, those things, it might carry some kind of residues. Of course, you do a lot of purifications at a DNA isolation level, but nevertheless, it might carry some inhibitors as a part of the sample. It should be resistant to the inhibitors. This is some of the criteria for selecting the so-called cyber green, right? You pay attention this couple of slides. It's coming here. The principle behind cyber green based qPCR. This is very important. So, what's happening is, when the start of the reaction, you have a template DNA which is by and large double-stranded DNA, right? And you have all other ingredients, and you added the dye here. Okay? At the 0 time point, you have everything together. You tell me what is the status of the template DNA? It is double-stranded, correct? And in terms of quantity, how much of double-stranded DNA is here? It may be around 30 nanograms or 15 nanograms. All of you know how much of starting DNA you use in PCR mixture? It is some nanogram level, and most often we use around 30 to 50 nanograms, right? So, remember this point. Now, what happens in the first cycle? The double-stranded DNA is converted into 2 single-stranded DNA, right? And the first cycle, what happens is, the polymerase chain reaction started, and it becomes 2 double-stranded DNA after the end of your first cycle, correct? Now, in the process, what has happened here is, the specific property of the cyber green is, this dye can go and intercalate in the minor grooves of DNA, double-stranded DNA. You remember this point, right? This is a unique property of cyber green dye. It has got a very specific affinity for the minor groove of double stranded DNA, right? So that means every cycle is passed, the freely available cyber green in the PCR mixture is going to intercalate in double stranded DNA, which is going to increase its number with every passing cycles. Is it clear to all of you? Freely available cyber green with every polymerase chain reaction step, there is an accumulation of double stranded DNA. The cyber green is going to intercalate. So what is the net result here? Whenever the cycle is completed, you are going to end up with a double stranded DNA which is already intercalated with cyber green. Simple. Second cycle, it becomes 4 strands. What's happening here? The unbound dye in the PCR mix with every passing cycle is getting depleted, correct? So in other terms, the growing double stranded DNA, I wouldn't call it as a growing, it's a replicating double stranded DNA is going to capture this dye after every cycle passes, right? So in a solution, the unbound dye exhibit very little fluorescence. That is another interesting property of this cyber green or this dye. Practically there is very little or no fluorescence when it is not bound to double stranded DNA. It gives excitation after binding to the minor groove of your double stranded DNA. So that means initially the fluorescence is very less. As the cycle increases, there is a progressive accumulation of double stranded DNA. When the double stranded DNA is formed, it is going to get this cyber green from the PCR mixture, correct? So cyber green binds to the minor groove of double stranded DNA helix. After annealing of the primer, dye molecule can bind to the double stranded DNA. DNA binding results in a dramatic increase of cyber green molecule to emit light upon excitation, right? Suppose when you reach around 20th cycle, just imagine 2 power 20 targets are created, or the template has released 2 power 20 targets, it depends upon the primers, correct? And that much quantum of your double stranded DNA is already having cyber green intercalated in minor groove. Is it clear to all of you? So when the double stranded DNA is converted into 2 single stranded, then again it is converted into 2 double stranded, the dye which is unbound in the solution is going to come and attach with the minor groove, right? And as and when the cycle progresses, there is a progressive accumulation of cyber green, right? The progressive increase in the fluorescence as amplicon accumulates, right? And intensity of fluorescence is measured by spectrophotometer. The question here is, how many of you have seen the qPCR results, right? I'll show you the picture here how it looks like. How do you visualize? I told the reactions can be monitored. What do you mean by monitoring the reaction? What is the indicator of the PCR is going on? Anybody? Graphs will come, right? So the graph or the raising curve is an indication of something is going on in the tube. Whether you ever realized why the graph is increasing, what is contributing to the raising graph? Anybody? Pardon? It is a fluorescence. There is a progressive accumulation of dye is happening, and remember 2 power 19 the cycle and 2 power 20th cycle. What is the difference between these two? In 2 power 20 the fluorescence is doubled than what it was at 2 power 19, right? So that is in nutshell about cyber green based qPCR technology, right? It comes with a kind of disadvantage. The disadvantage here is the cyber green does not recognize what is there in terms of sequences. whatever double stranded DNA comes in its way, it is going to intercalate. Suppose if you are amplifying a gene in insect, for example, COX, and you want to do this technology, right? By chance, there is some contamination in your PCR mix and something else is also amplifying which you are not realizing. So what happens here? That also parallelly amplifies. So your cyber green is going to bind there also. So that's the reason it is called non-specific. Is it clear? Why it is non-specific? Because cyber green has got a very high affinity towards double stranded DNA, no matter from where the double stranded DNA comes, right? Maybe a kind of your target, real target, maybe a false amplification happening, false positives, it's quite possible. And cyber green does not recognize what is getting amplified, right? So that is a disadvantage with this technique. So of course, there are plenty of advantages. That's the reason we all use this. Very easy to produce, very cheap, easy to handle, of course very sensitive and very effective. And the disadvantage is non-specific. If this is clear to you, I will move on to the next one. Is it clear? If anybody is not clear, you please let us discuss maybe the end of the presentation, right? Now let's move on to the probe-based quantitative PCR. In dye-based quantitative PCR, you added a fluorescent dye in your PCR mixture, correct? In this particular technology, instead of a dye, you are going to add a probe. What do you mean by probe? Probe is nothing but very short nucleotide sequence, right? All of you know the primers, correct? It is nothing but primer kind of stuff. You can think like that. It's not exactly the primer, but it is a nucleic acid fragment only. It is specific to the target. Suppose if you are developing a qPCR based quantitation of some genes in insect. I'll put it like that. That's some gene I told, maybe one gene, any one insect gene you take, COX I will tell, right? And COX is a target here. And COX gene has got a length. Is it clear to all of you? And there are two things. One is primers. You have a COX specific primer. And what is probe here? Probe is nothing but a short nucleotide sequence which is complementing to the same gene which you are targeting in your PCR. In other terms, you are having two. Primer is also a probe only. If you think deep, primer is also a probe only. Here you are going to use, I would put it like this, two primer kind of stuff for amplifying. Primer is going to mediate the amplification. The probe is going to go and bind to the target. This you must understand. The probe is the one which is added in the PCR mixture in addition to the primers. The only difference is you are going to decorate this probe by adding some chemicals, right? The probe-based qPCR relies on the sequence-specific detection of a specific PCR product, sequence-specific detection. That is completely different from the dye-based one. In dye-based, you added a chemical which is going to intercalate in the minor groove of double-stranded DNA. But in the probe-based one, the probe is going to bind with the target, right? Unlike cyber green based qPCR method that detect all double-stranded DNA irrespective of the target, the probe based qPCR utilizes, just now I told you have to decorate this probe by adding some chemicals. How? You are going to label the probe using a fluorescent chemical, also called as fluoroprobe, right? The probe is now decorated with a fluoroprobe, and it is target specific probe, resulting in increased specificity and sensitivity. Let's see how really it works. We have a number of choices right? For example, Tachman probe, molecular beacons, Scorpion primers, hybridization probes, these are some of the examples. And of course, the Tachman is the most popular. It was invented sometime in 1996, right? This is a Tachman probe. What you see here is the probe. Probe is specific for the target gene or whatever target you are addressing in your project, correct? So this probe is decorated by two chemicals. One is called a fluoroprobe, indicated as R, and another chemical is quencher. In few slides you will come to know why this setup is needed in this particular technique. You have a probe. On one side you have a fluorescent, in the symbol language fluorescent dye is attached in the 5 prime, and another side you put the quencher. I hope you understand what is quencher. Quencher is something which is going to suppress the fluorescence. It's a literary meaning of quencher is it will suppress the fluorescence emission, right? So now this is the probe, right? And it is specific for the target gene what you are addressing in your project. So what happens? In a mixture, you are adding this probe as one of the ingredients in qPCR, correct? In addition to the rest all. So now you set up the PCR mixture, PCR reaction, right? So what happens? A single stranded will become two stranded. Since it is a two stranded, what is the property of a single stranded DNA? Whatever single stranded molecule is there, they will go and complement with each other. So what happens? The probes, which is freely floating in the PCR mixture, right? They have complementarity with the target, and this probe is having two chemical attached to it, one on 5 prime side, another one on 3 prime side. They will go and bind to the single stranded template now. Why the single stranded template here? Because it is already subjected to denaturation. Is it clear? Now probe and the single stranded DNA is what you call complementing with each other. That is what it is shown here, correct? It is here, somewhere here, right? Now what happens? Please read it. When intact, the fluorescence of the reporter is quenched due to its proximity to the quencher, right? The probe hybridizes to the target because the target is made into a single stranded because you subjected the DNA to high temperature, that is 94 to 96 degrees Celsius, what is called as denaturation temperature. Is it clear? Now the double stranded DNA is specific. Here comes the twist. So you also use an enzyme for mediating the polymerization. What is that enzyme? Taq polymerase enzyme. This Taq polymerase enzyme has got a very, very unique trait or property. That is, it's written here, 5 prime to 3 prime exonuclease activity. Is it clear? So this particular activity is exploited here. How? Let's see in future slides. Now what happens? The target is already bound to the single stranded DNA, and remember, parallelly the primary is also going to bind. It's a very dynamic system. It's not one after another. It's all parallel events. Correct? It's all parallel events. So you have a single stranded DNA. A target is bound somewhere. The primer goes and bind here. Right? You have a quencher on the 5 prime and a reporter on the 3 prime. Right? Now what happens? The primer, where it bound, immediately the polymerase enzyme comes and start extending it. As it extended, somewhere on the way, it is going to encounter the target, which is already having reporter and quencher, right? So now, the exonuclease activity of your 5 prime to your Taq enzyme is going to strip this Taq, what do you call the probe, what is already bound with the single stranded DNA. That's what exactly happens here. So it removes the quencher, it also removes the individual nucleotides and also the reporter. right, with the help of a specific activity what is called as 5' to 3' exonuclease activity, right. Now, in a pictorial it is something like this. On 5' you have the fluoroprobe, and 3' you have the quencher, and it's a double stranded DNA in the start of the reaction, and everything is freely floating in the mixture. Now what happens when the temperature is the denaturation temperature? So the two single stranded DNA is formed, and your probe is already attached somewhere here, right. Now what happens? You continue to incubate. So now the primer comes into picture. Right. You can see here the primer is binding somewhere here on the complementary template strand, and it is going to just follow the diagram, the DNA polymerase, which is given in green color, and that is going to extend the primer, and when it extends the primer, goes beyond it, and it is going to what do you call, just see here, the elongation takes place. During elongation step, it is going to remove the so-called quencher and also the reporter. So when it is, just see here, it is started removing that, and the 5' to 3' exonuclease activity of the polymerase removes the fluoroprobe and probe nucleotide and also the quencher. All the three will be separated from each other. So they will come into the solution as an individual entity rather than a complete probe, right? Because the enzyme activity has removed them, and also they disintegrated the entire probe into small pieces in a simple language. So what happens here now? When they were intact, the fluorescent probe and also the quencher is suppressing the fluorescence activity of the reporter or the chemical. Now it is removed from each other. Now the reporter is not under the quenching activity of the quencher. It started giving the fluorescence, right? So every cycle, passing cycle, what happens? The intact probe is disintegrated. Remember, the intact probe is not giving any fluorescence because of the quenching activity of the quencher, right? Every cycle, what happens here? There is a depletion of the intact probe that is converted into the individual components of this probe, like reporter separately, quencher separately, and the intervening nucleotide separately. So what is the net result here? There is a progressive increase of free fluoroprobe in the PCR mixture. So every passing cycle would add the equal to the previous quantity of what you call the free fluorescent chemical into the PCR mixture, right? So the fluoroprobe emit fluorescence after its a detachment from the quencher. So that you should understand. That's a big difference here as compared to cybergreen based qPCR. In cybergreen based qPCR, it is just opposite to that. There, the dyes are bound in the what you call minor grooves. But here, there is a depletion of the intact probe, which is otherwise not giving any fluorescence. After the what you call the reaction is completed with the help of 5' to 3' exonuclease activity, there is a progressive increase of free fluoroprobe in the PCR mix, which would add to the so-called fluorescent emission in PCR mixture. Is it clear? Yeah. So now what happens when the cycle is around completed? So you have so much of free fluoroprobe, which is already available in the PCR mixture, that can be monitored by the camera kind of stuff, which I already told, the spectrophotometer, which is kept on the top of the PCR machine to monitor what's going on. And the fluorescent intensity is captured by spectrophotometer. The 5' to 3' exonuclease activity of the polymerase removes the probe and nucleotide and quencher, and every amplification adds free fluoroprobe in the PCR mixture, thus increasing the overall fluorescence after every passing cycle. Now the question is how the chemical which is going to give you fluorescence is quenched by the chemical. Let's take some examples. There are two mechanisms are reported. One is called FRET quenching, another one is non-FRET quenching, right? Maybe that the duration of this presentation may not permit me to elaborate on that. There is a great deal of information is there already. You please try to find out the finer differences between these two, but in nutshell, I will tell you what's really happening. One thing is sure, as long as the quenching, as long as the fluorescence probe and the quencher is together, separated by few nucleotides, the fluorescence is under the quenching activity of the quencher. That means there is no fluorescence at all, right? And the mechanism of quenching the fluorescence is two mechanisms. One is FRET, another one is non-FRET, right? FRET stands for fluorescence resonance energy transfer, right? So this is a FRET quenching. What happens is energy passing upon light hitting on the fluorescence, dipole to dipole interactions. Please read during the course of this training here. Let us interact on this again. And this is one form of quenching the activity of fluorescence, right? This is happening after light passing through the fluorescence, right? Non-FRET quenching is nothing but when they are together, separated by few nucleotides, fluorescence or non-fluorescence. Is it clear? What I mean, the fluorescence cannot emit light as long as the quenching chemical is together or separated by few nucleotides, right? This is non-FRET quenching. So how these two are separated? How this quenching chemical and fluorescence chemicals are separated from each other? Just now I told 5' to 3', exonuclease activity of the very same enzyme what is used in DNA polymerization. That's the beauty of the whole technique. You are not adding something new here. It is the same enzyme which does the polymerization, the same thing which is happening for removing the probe molecule, which is composed of three chemicals. One is reporter, quencher, and the nucleotides, ATCG, right? So non-FRET quenching is exploiting the property. As long as these two molecules are close proximity, fluorescence cannot happen. When they are removed from each other, it emits fluorescence, right? Let's take some examples of well-known fluorescent chemicals which is reported and very popular in qPCR. FAM, JO, TET. I try to remember this. FAM stands for tetrachlorofluorescin. Fluorescin amidites. TET stands for tetrachlorofluorescin, and JO is a kind of chemical 4,5-dichloro-2,7-dimethoxy carboxyfluorescin. This is not the end of it. You have quite a lot of fluorescent molecules in the literature. I just brought the most popular one, right? And this is the quenching molecules, right? You might have heard about these quenching molecules. You have THAMRA, you have BHQ, and you have MGP, right? It's all only acronyms just to confuse us. They are nothing but tetramethyl rhodamine, that is THAMRA, and BHQ is nothing but black hole quencher 1, right? Remember this, black hole quencher 1. And have MGP, that is nothing but minor groove binder, right? So your cyber green is also coming under this category only, right? Minor groove binders. So this is an example for quenchers. The previous one was the example for reporter. So remember, the common name is a reporter. So you have some choices here, you have the quenching choices here, right? Now let's come to the advantage of this probe based. This is clear to all of you, the difference between the dye based qPCR and probe based qPCR. Will you be able to write if I ask you to write a paragraph about it? All of you? Let's be very frank. If it is not, we'll retake once again. Anybody is not understanding or any difficulty in understanding? Is it clear to all of you? Sure? Can I move ahead? Good. See, pardon? You have to elaborate on your own. Maybe form groups. Maybe half an hour time will give, right? So a lot of chemistry, physics and chemistry are involved there. Light, fluorescence. You please read that. Okay. So FRET and non-FRET quenching. Finally, what happens is the fluorescence activity of the chemicals what is given here is quenched with the help of the quencher. That is explained by two mechanisms. One is FRET mechanism, another one is non-FRET mechanisms. It's not a big deal. Just you need to understand dipole-dipole interaction. If you understand that, you got the answer, right? You try to understand that dipole-dipole interactions. That is something to do with the energy transfer from fluorescent chemical to the quenching chemical. In the process, these two molecules are something like dipole-dipole interaction that leads to the so-called fluorescence happening to the fluorescing probes, right? Non-FRET is quite easy. When they are together, the fluorescence activity of the fluorescent molecule is completely quenched. Quenching means suppressed, right? It is stopped. So once the quencher is removed, it fluoresces. It's like it's a kind of molecular switch you might have studied, right? Off position when they are together. So when they are removed, the fluorescence is on position. It's going to give you fluorescence, and that is being captured by the spectrophotometer. Now, these are all some of the examples I brought here. The advantage of probe-based, you yourself can tell, extremely specific, because it's a double detection system. You have a primer-based one and also the probe-based. Remember, these two are specific to the target. Is it clear? The probe and primer, it cannot be same, by the by. Somebody was asking in the previous class, can the primer and probe be same? It cannot be same, because if it's same, it doesn't work actually, because they compete for the same location for complementarity, isn't it? So they need to be separated from each other, right? And in general, the primer binding is upstream of the so-called probe binding, so that the TAC enzyme encounters this probe in its way of extending the primer. Is it clear to all of you? Right? When the TAC enzyme is adding the nucleotide, and after certain addition, it should encounter the probe, so that the probe is removed and new nucleotide is added. Remember, end of the day, the sequence is same only. The only thing is, it removes the pre-existing probe there. In its place, it is going to add the new nucleotides. Two enzyme activities are involved here. One is 5' to 3'. Exonuclease activity is going to remove the pre-existing probe. At the same time, the same molecule is having another activity, polymerization, that is going to bring another nucleotide. Maybe these two nucleotides are same only. The only difference is the probe and the quencher and the fluorescent dye will not be there any longer. That is a secret. If you understand this, then you understood this probe-based. Nothing more than that. 5' to 3' exonuclease activity is the key here, because that is the one which is going to remove the quencher and fluorescent togetherness. Correct? It is going to separate them, so that the net fluorescence which is coming from the mixture is going up every passing your polymerase reaction steps. Right? Yeah, the advantage already told is specific, and multiplexing is possible here. And disadvantage is, of course, you have to have a specific nucleotide sequence which is going to serve as a probe for you. And most important thing is, you have to order a company to convert the probe into the so-called what do you call a setup which is having fluorescence and also quenching activity. So that's some company will help you. You have to provide the nucleotide sequence to them. So they synthesize the probes for you by adding appropriate the dye which you have to select, which I already told. It may be a FAM, it may be TET, anything which is of your choice, and also the quenching. So the quenching chemicals, maybe TAMRA or PHQ. Just now I told any one of them. They will add these two, and they will supply to you the probe. It is much like the ordering the primer. You order primer, after a couple of weeks, the primer comes. Something which is very similar to that. There is no big difference. Okay, please remove this confusion. Probes are made to order, and it is supplied by a company, right? And that you are going to keep in your lab. And remember, for every target, you have to make this probe. That is the kind of, it's expensive, and it is hard to sequence sometimes, and of course, somebody reporting the false results also. But nevertheless, this is extremely specific for qPCR applications, right? Yeah. Now, I'll take one slide just to tell you this particular one, that is a reverse transcriptase PCR. The acronym RT-PCR is also used for reverse transcriptase PCR. Here the target are by and large RNA generated targets, right? I think all of you must be knowing RNAs are converted into a kind of DNA, what is called as complementary DNA, also called as cDNA, and these cDNAs are used here to do that particular step, converting this RNA into cDNA. We add an enzyme, what is called as reverse transcriptase enzyme. For this purpose only, this particular tool is called RT-PCR, reverse transcriptase PCR, right? And the cDNA is used as a template, right? Yeah, this is in pictorial depiction of what's really happening. I'm just tempted to tell you two variant of this particular technology. Remember, qPCR applications, I already made a mention here for gene expression studies. They are one of the most handy tools, right? Gene expression, the moment I talk about gene expression, you have to talk about RNAs only, right? So what you do, maybe in your case, maybe you make the plant infested by an insect, then the post-infection you take samples, maybe the different time points, then you extract RNAs, correct? So what we are doing is, when the insect is infesting a host, what and all changes which is taking place in the host, if that is an objective of your experiment, then you need to handle RNA in your experiments, right? So you have isolated RNA. Now the thing is, you want to quantitate how much of RNA is there in your sample, right, in that particular time point. So here comes the so-called real-time based quantitation of the transcripts. Okay, so here normally we go for two same protocol, but nowadays we have something like one-step RT-PCR and two-step RT-PCR, right? In one-step RT-PCR, everything is in a single tube, right? It all kit based only. It has got all ingredients which is needed for PCR, and also an enzyme which is needed for converting this RNA into cDNA, right? All in one. This is a single step RT-PCR. It's not a big deal, right? Conventionally, people do a two-step RT-PCR also. That means the applications, it varies. Let us not forget that. For example, if you want to preserve your cDNAs as a separate, what do you call, a chemical in your lab, or a kind of biological material in your lab, you have to necessarily go for two-step RT-PCR, right? In a single-step RT-PCR, there is no cDNA left because everything is in situ there, and you are straight away coming into gene expression analysis. But in the case of two-step, what you do, initially you convert your RNA into cDNA, and that is kept as a separate biological material, and you take an aliquot of that and you set up qPCR here. So this is two-step RT-PCR. This is clear to all of you? Single step and two-step. In single step, everything is performed in a single tube, and that includes the conversion of RNA into cDNA, right, and also the enzymes and salt and buffer which is needed for PCR, and also the transcriptase, the transcription, reverse transcription function. So everything is in a single cocktail, right? So this is used by and large used in the case of high throughput gene expression analysis. People do not have time for separately converting into cDNA, preserving in their deep freezers, and take the aliquot later on to do something else. So it's simple. One step and two step has got plus and minus. It all depends upon your experimental setup, what you really want out of this experiment, right? The requirement for qPCR, of course, you need a machine which can perform this PCR and also which can monitor the reactions. And these machines are popularly called as real-time PCR machines or qPCR machines, right? And of course, a template. We need to have a template is nothing but the target on which you are working. In your case, maybe an insect target, maybe a host target, right? And it may be a RNA or it may be DNA. It all depends what you really have in your experiment. So DNA, most of the time in the case of estimation of certain targets in the environment, right? That is DNA. RNA, by and large, it is for gene expression analysis, or you want to quantitate the transcripts upon certain treatments. So you have RNA as a a template here. Remember, RNA having said that is a template, but they are not directly going as a template. You need to convert them into cDNA. Please understand like that. cDNA is the final target here. Of course, primers. Primers are so important here, and you need to have the knowledge on primer designing and also the kind of activity associated with primer designing and validation. I think there may be a separate class Dr. Subramaniam has already made, so somebody will deliver a class on that. So primer designing is the most critical step that decides the outcome of your experiments. By the by, I would like to ask you one question. In CyberGreen, I already told it is having non-specific reactions. Can we control the non-specific reactions in CyberGreen based qPCR? What is the way out? The only way out is you have very, very specific primers, and it is not having any non-target amplifications. So that is the reason why CyberGreen is famous, because we have better what do you call online platforms to design primer, and those primers are really working wonderfully well. So people are comfortable with the so-called CyberGreen. The reason is the primer designing is what do you call not a big deal nowadays. We have a number of platforms available online, and you can formulas have been devised to come to a kind of annealing temperature which is going to amplify only the target. So it's quite possible nowadays. So have a specific primer that would solve the non-specific problems what you may encounter in CyberGreen based qPCR, right? And Tachman probe, of course, if you are too tech savvy and you want to go for very specific detection, then you need to have another target, another sequence which is designed from the same target, and you are going for probe making by ordering to a company to convert this sequence into a kind of probe. That is possible. And DNTPs, of course, this is again available commercially, and you have other ingredients like magnesium chloride, we have Taq DNA polymerase, and of course buffer systems, and everything is commercially available except perhaps the template and also the primers and probes that is in your hand. Rest all commercially available. And cycling condition, it is nothing but the same PCR conditions. There is no big difference. Same. You have a denaturation temperature, you have a kind of annealing temperature, you have an extension temperature, and of course you are going to repeat it for n number of cycles, right? And remember, every ingredient what you use in qPCR has got a shelf life, and after every passing cycle there is a depletion of those resources. That's one of the reasons why we settle down for 30 or 35 cycles at the maximum, right? You cannot do this cycling for 100 times, right? You just sit and work out. If 100 cycling is possible, what would have been the quantum of PCR product, right? You just sit and just work it out, 2 power 100. That's going to be enormous astronomical number, right? You'll be really surprised. You just sit and calculate how much product is going to come out of the PCR tube. That's only theory. It is not possible at all, right? And of course, come to the visualization of PCR results, qPCR results. This qPCR results can be visualized, that the visualization itself is divided into 4, right? To start with linear ground phase, I already told there is no ground fluorescence either by Tachman probe method or CyberGreen method. Why? In CyberGreen, the free dyes are less or no fluorescence. Once after binding with the double-stranded DNA, only it is going to give you emissions, right? Number one. Tachman probe, the story is same again because it is quenched, right? So initial ground phase, absolutely no fluorescence or low fluorescence which the machine cannot see. There may be some little ground fluorescence because remember, you are adding 15 nanogram of double-stranded DNA where theoretically the dyes can go and bind, especially in the case of CyberGreen method. But remember, that binding will not give you any tangible fluorescence to the machine. So there will not be any graph at all. It will be lag phase. The second one is early exponential phase and the linear exponential phase and the plateau. Let's see. This is the linear ground phase, right? So this is typically the linear ground phase, right? And in the first phase, PCR is just starting. Fluorescent signal has not risen above the background, right? The machine is unable to capture any any lights or any fluorescence coming from the setup. The second phase is early exponential phase. Is so critical for us. The second phase is where the fluorescent signal just rise significantly above background. The cycle at which occurs is called cyclic threshold level. This is one of the indicator of the so-called qPCR results. You might have heard about CT values or CQ values. You know, what do you mean by CT value or CQ value? CT or CQ value is nothing but this particular point, right? The machine started seeing some fluorescence from the reaction mixture. That is displayed in the form of raising curve, right? When the curve started raising, the raising curve is what is called as the CQ value, that is above the background. And the linear exponential phase is where the intensity of fluorescence doubles every passing cycle, right? That is mostly in the log phase, right? That is somewhere it is here. And of course, with the passage of each cycle, when number of cycles are reaching around 30, 35, there is a depletion of every ingredient in PCR, including the dye, right? There is a shortage of all inputs which otherwise would have contributed for the accumulation of double-stranded DNA, and there is a shortage, and the reaction automatically comes to standstill, right? Because the enzyme loses its activity, there is no free DNTPs, there is no fluorescence, and template DNA may be there, but you may not have the other things which is needed for accumulating the further double-stranded DNA. It is not there. This is a plateau that what is called as the last phase is when the substrates are exhausted and TAC enzyme loses its activity, and the fluorescence signal will no longer increase, right? It takes a kind of straight line, right? That usually that happens after 31 to 32 cycles. By and large, 34th cycle is the last point in most of the qPCRs nowadays. And come to the data analysis, and I already told the baseline amplification plot. You will encounter this kind of terminologies in literature. Baseline of amplification plot, that the initial cycle of PCR, there is a little change in the fluorescent signal. That lasts even up to 15 to 20 cycles. You may not see the curve raising most of the times, right? So it goes like this only. That is called as lag phase, right? Suddenly, after 18th, 19th, 20th cycle, sharp increase in the curve goes up, right? So that is a starting point for your CQ values, right? And an increase in the fluorescence above the baseline indicates accumulated PCR product. And threshold line, a point at which a reaction reaches the fluorescent intensity above the background. It is set in the exponential phase of the amplification for most accurate readings, right? And CQ threshold, CT, the cycle at which the sample reaches the threshold level. CT values are 40 or more means no amplification and cannot be included in the calculations. If somebody is telling I have a CQ value of around 34, you can easily understand that there is nothing happened in that particular sample, right? And another terminology you encounter is a melting curve, that is the change in the fluorescence when the double-stranded DNA incorporated dye molecule melt into a single stranded as the temperature increases. Remember, the dye is intercalated in the minor groove, and the temperature, when you subject that stuff into around 80 or 85 degrees Celsius, it becomes true double-stranded DNA. There is a sudden drop in the fluorescence. That is called as melt curve analysis. So important in the case of cybergreen based qPCR, where you have to get a single melt peak, right? If you get a single melt curve, that indicates that the primer is the most accurate primer for the target, right? If you have multiple targets, multiple amplification took place that contributed to overall cybergreen fluorescence, you will see a multiple peaks here. In this particular curve, everything is single, right? That means the primer has got only one target. Upon melting, it created a single melt curve. This is also so important in qPCR analysis, right? When the double-stranded bound cybergreen dye is heated, a sudden decrease in the fluorescence is detected at the Tm value. The Tm is nothing but melting temperature of that particular target. Yeah, this is, we'll come to that in the next slide. Now, can I continue now for another 10-15 minutes? Yeah, I'll quickly rush through because there is a separate talk is there, but I will only highlight the key points. The gene expression analysis, which I already told, there are two ways of handling this particular tool. You want to know whether the gene is upregulated or downregulated, or in other words, induced or repressed. You can analyze through this qPCR, right? And there are two terminologies are very popularly used. One is absolute quantitation, and another one is relative quantitation, right? In absolute quantitation, it's quite simple. Suppose you know the target, for example, a gene in an insect, you know it already. So what you do, you develop a standard curve, you plot the concentration of that particular gene in terms of nanograms or picograms versus the CQ values what the machine is going to generate. It's nothing, you set up a PCR reaction with the differential quantities of your template DNA. Is it clear? If it is RNA, you convert into cDNA and make the cDNA as a template. So what you do, you start from theoretically very low quantity, maybe a picogram level, you go up to nanogram level and make a different concentrations, maybe 1 picogram, 10 picogram, 100 picogram, 1000 picogram, you go up to maybe let's say around 10 nanograms, right? Or even up to let us say 100 nanograms. Right, and each one of them is subjected to PCR. Right, now what you do, you set up a qPCR. What happens here, if I have very high quantity of initial template DNA, what would be the CQ value? Yeah, the CQ value comes early when you have more template in the initial phases. Is it understandable to all of you? Is it clear to all of you? If you have a more of a template, the CQ value will come early. If there is no template, CQ value will never come, right? So remember this point, isn't it? So using this particular criteria, you can make a standard curve, right? Because the CQ value or CT value is what do you call value in terms of number, it looks small. For example, if you are getting a CQ value of around 5, that means you have so much of template, right? Higher CQ value indicates low quantity of template DNA or template cDNA. Remember this point. So this is exploited in making a standard curve, is something like this, right? That means for every given concentration of your cDNA or template DNA, what is the CQ value? Once this is known, your test material can be analyzed by using this standard curve. That would tell you how much of target was there at the time of your experiments. Right? This is nothing but any other spectrophotometer reading only. You have a standard curve and decode the values or the quantity based on what you call the standard curve method. Right? So this is absolute quantitation, and another one is what is called as this is something like this, require a construction of an absolute standard curve for each target. Standard curve is based on the serial dilution of the sample with a known copy number, and the CT of each standard is plotted against a logarithm of a known concentration, and standard curve is then used to estimate the concentration of unknown your test material. Another one is relative quantitation. This is very popular in the case of gene expression analysis, right? Here in absolute quantitation, you will come to know how many copies of transcripts were there that can be estimated precisely using this technique. In the case of relative quantitation, you will be telling whether the gene X or Y what you are analyzing, how it is behaving in a situation where the host is infected by an insect or maybe whatever conditions. This is relative. Relative to what? Relative to a reference gene. Right? You always remember you have to keep one reference gene in your analysis. Right? So I'll take you to a small information which I could update my slides. Very often we encounter a problem, what should be the reference gene? Remember, reference gene is the one which is not affected by the conditions at all, right? No matter whether the condition is, I'll take a small example. If you are having a plant which is infested by a sucking pest, there may be a great deal of changes are happening in the host gene expression, and these reference genes are selected. Those genes are not affected at all. whatever may be the condition, their expression is constant. This point you keep in your mind. Those genes are taken on board as a reference gene. Yeah, yeah. Yeah, you take that. Yeah, choice of housekeeping genes. This paper I could encounter from this in physiology. It's a very nice paper. What I could see is they have come out with a very comprehensive review of all reference gene whatever is used in insect qPCR applications. Right? The title goes selection of reference gene for normalization of RT-PCR data in gene expression studies in insects. A systematic review. It's really a systematic review. I could see every order of insect they come out with a choice of genes what is published by different scientists all over the world. Right? I just showed only a glimpses of this coleopteran insects, and the table goes and goes and goes up to covering all the insect orders. Right? You please read this paper. It's a Frontiers open access paper. And one thing I found very interesting here is actin gene. Most often we use in the eukaryotic system actin gene as a reference gene that is very popular in most of the insect order. This particular gene is there. Starting from the first one to last, actin gene is there. Right? Anyway, nevertheless, you have to standardize the reference gene. Whatever conditions you are using, whatever setup you are using, that is a mandatory. You cannot go with the literature. Right? And this is the difference. Absolute quantitation determines the expression level of the in absolute numbers or copies. Relative quantitation determines the fold changes in the expression between two samples. This is the most important point. Suppose if you are analyzing a particular insect responsive genes in the host, you need to have a reference gene. In relation to the reference gene, how those insect responsive genes are behaving. That in terms of fold change. Right? Fold change means how many fold expression in relation to the reference gene you got it in your sample. It may be, remember, upward and also downward also. Right? That is the most important point. And come to copy number estimation. This is an updated version here. Now we have an online support. Straight away you get into this particular website, scienceprimer.com. That gives you this particular page just to feed the data into this particular online portal. That is going to give you how many copies of targets are there. Right? So it's quite simple. Only thing is you need to find out X and N that you have to generate from your qPCR experimental data. Right? X is in terms of the quantity you got amplified, and N is what is called as the length of the double-stranded DNA amplicon, the target length. On these two input data you have to generate from your experiment. You just substitute in this online portal. It is looking something like this, and when you just submit it, automatically the copy number comes here, readily displayed right in front of you. This is one of the very handy tools for what is called as copy number calculator for real-time PCR experiments. And some consideration, and absolute quantitation is quite easy and simple. Only thing is you need to standardize, you need to produce a standard curve, and using online portals you can quantitate how much of target was there. In terms of relative quantitation, I already told you, you need to have a reference gene. And here a small thing I would like to highlight is so important. In relative quantitation, there are certain terminologies you need to keep in your mind. One is calibrator, another one is test, and another one is target and reference. Let's take one small example here, maybe from the host side. Calibrator is nothing but a plant which is not subjected to the experimental treatment, maybe an insect attack or maybe a pathogen attack or maybe some chemical. Right? It is what is called as absolute control. Right? Test is the real experiment what you are doing. Right? That is the inoculated or infested plant. Keep in your mind. And target and reference. Target is the gene which you took on board. Through that you are trying to answer a biological question. Right? That may be a kind of a gene which is responsive to insect attack or infestation, or maybe which is responding to a kind of insecticide experiment you are doing. Right? And reference, which I already told you, the one which is not affected at all under any conditions. Right? In all real-time PCR where you want to do this relative quantitation, you have to satisfy this checker, the quadrant checker. You need to define the calibrator, you need to define the test, you need to define the target, and also need to define the reference gene. And all the data it was taken on board, and we use any one of this technique to come to a conclusion about the pattern of gene expression in that particular experiments. Remember, in literature you will find a LEVAC or double delta CT method is the most popular method. We have a delta CT method using a reference gene, and also FAFL method. These three methods, I think we have an exclusive lectures on that. They will be feeding you more on this, right? So, but nevertheless, what is more important is this particular a checkerboard. You have to write and you have to put on black and white what is that you want to do. And what do you mean by calibrator here? What do you mean by test? What is the target and what is the reference gene? Without doing that, you cannot proceed into the next level. Of course, for relative quantitation, it's quite easy because you need to have kind of no need to know the starting concentrations. Very useful for entomology research where the plant or pest gene expression due to presence or absence of each other's factor can be beautifully worked out. This is one of the biggest advantage of having this kind of tools, right? I think that's the last slide, if I guess. Yeah, applications, of course, a number of applications are there, which I already told. The first and foremost application of qPCR is gene expression analysis. You have detection of mutant genes, you have copy number measurement, absolute quantitation I told. Of course, from the microbiological point of view or pathogen point of view, if insect is affected by a virus or pathogen, you can estimate the load, viral load or pathogen load in those insects, right? And the single nucleotide polymorphism analysis you can do, and so many other applications are also there in qPCRs, right? And these are all some of the machines which is available in the market for performing qPCR. Light cycler from Roche, we have ABI Prism 700, and these are old slides, and we have a lot of new machines are there. CFX 96 from Bio-Rad is the most popular ones going on in many laboratories. So please Google it out, you'll get more and more information on this. So with that I conclude. I think I exceeded by 20 minutes, I'm sorry for that. And with that I'll conclude this presentation. If you have questions, let us try to have some interactions on this. Maybe we'll take another 5 minutes maximum.
Questions?
Actually, we planned as a 3-pronged lecture. This lecture is planned as a case by